So we've talked about cell division and the mechanism by which cells control the division of the nuclear contents, i.e. the DNA. We've talked about the details of meiosis and how chromosome number is controlled across the generations by the generation of haploid gametes that have half the genetic content of the germ cells that we began with. And you might have wondered during that discussion whether mistakes ever happen during that process. And the answer is yes, mistakes do happen during that process. So in this video, what I'd like to do is talk a little bit about those mistakes, those errors that can occur during cell division, with a particular focus on the errors that occur during meiosis, because obviously meiosis is that mode of cell division that is generating gametes. So this can have particularly disastrous consequences in the context of human reproduction. So we're going to talk about a couple of different types of errors in meiosis. We're going to talk about errors that occur during recombination or crossing over, when DNA molecules are actually breaking and exchanging genes. And then we're going to talk about errors of segregation, where there's a failure of the spindle checkpoint during meiosis, either meiosis 1 or meiosis 2, and therefore the chromosomes don't segregate properly in that cell division. But we're going to start with recombination errors. So the first type of recombination error that I want to talk about is what's referred to as non-reciprocal recombination. Okay, so what normally happens during meiosis, more specifically meiosis 1, prophase 1, is that the homologs, the duplicated homologs that were produced in S phase, remember we had each chromosome copied to produce two sister chromatids held together by our cohesin proteins. That's a duplicated homolog. Those duplicated homologs that match one another are paired up, and we see a crossover event that can be described as reciprocal, meaning that there's a specific breakpoint that's introduced on the DNA at the same position on each of those non-sister chromatids, and then there's that exchange, so that's swapping of genetic material. And under normal circumstances, those breakpoints are positioned in the same place along that genetic sequence, and equivalent bits of DNA are exchanged. So that's what's referred to as reciprocal exchange or reciprocal recombination, right? So that's the normal state of affairs. But what happens if the breakpoints that are introduced are not exactly aligned with one another? In other words, let's say that on this chromosome, we had a breakpoint introduced between these two sequences. Let's say these are genes B and C. But on this set of duplicated homologs, we had a breakpoint introduced up here instead. So not in the same place. And then we had the crossover event. So these bits of DNA were broken and they exchanged locations. And so now this leg of this chromatid is attached to this chromosome. This leg of this chromatid is attached to this chromosome. What would we get after that meiotic division, that first meiotic division proceeded? Well, if after that exchange, what we would see is that on this chromatid, we'd actually have a duplication of whatever this gene B is, right? So it would have had AB and then BCD transferred from the other chromatid. And by contrast, this chromatid would now have genes A, C, and D, and would be missing gene B because it got the original A that it contained and then C and D from the other non-sister chromatid. And so what we see is that this chromosome has suffered a deletion of a gene, and this chromosome has now achieved a duplication of this specific gene. Now, what's the consequence of this? Well, it depends very much on what that gene B is. If it is a gene that is not really subjected to very tight regulation in terms of how much of that gene is expressed and at what time points during development, then this may not be a very important event, and that may be tolerated very well. But if this is a very important, let's say, developmental gene, and the dosage is very important to maintain, then having an extra copy of that gene or a missing copy of that gene could actually be associated with disease. Because remember that what's going to happen to each of these sister chromatids is that they're going to go through with meiosis 1, and in meiosis 1 we're going to see these homolog pairs separated from one another, and then they're going to go through meiosis 2 where the sister chromatids are separated from one another, right, when those cohesins are cleaved. And so each of these chromatids is going to end up in a different gamete. So if we just write down what genes are going to be present, let's say these are eggs, right? This chromatid ends up in this egg, and genes A, B, C, and D are present. This chromatid ends up in this egg, and so this egg has A, B, B, C, D. This chromatid ends up in this egg and only has genes A, C, and D from this chromosome, and this chromatid ends up in this egg, right? So these are ova, or eggs, and we know that in the process of fertilization, they're going to be uniting with some sperm that is going to be ideally carrying its own set of correct DNA, and it's going to unite with one of these eggs, right? So if it unites with this egg, we can see that that new embryo that's formed is going to be one copy of gene B down, right? It didn't get one from this maternal parent, but it did get one from the other parent. If this sperm unites with this egg, then that new embryo that's formed is going to have one extra copy of that gene B. So again, depending very much on what that gene B encodes, we could see different consequences as a result of that either duplication or deletion of genetic information. Now we're going to see a little bit more about this idea of gene dosage effects and gene duplication and how that leads to variation across the genome, and we're also going to see in just a moment how that can be associated with gene families. But what I want to do now is talk about that second error in recombination that can happen, and that's an error where the wrong chromosomes get paired up in meiosis 1. So as we just saw under normal circumstances, what gets paired up in prophase 1 are the matching sets of homologs. So this duplicated homolog that has genes A, B, C, and D should be paired up with its partner homolog that also has genes A, B, C, and D. And you know that chromosome exists somewhere in the cell, but let's say that the cell makes a mistake and accidentally picks up and pairs with this duplicated homolog, the duplicated homolog that has genes E, F, G, and H instead, right? Now you might ask yourself, well, how can that be possible? Because these sequences shouldn't be similar at all. So how could the cell make a mistake like that? Well, it turns out that an incredible amount of the human genome is made up of repetitious DNA, so repetitive DNA sequences that can make mistakes like this pretty common and hard to detect at the molecular level. All right, so what would happen if these chromosomes were paired up and then we saw a recombination event between them? Well, let's say that the breakpoint is here between genes B and C on this set of homologs, and then the other breakpoint was between genes F and G on this non-homologous set of chromosomes, and then we saw exchange of that genetic information. Well, what we would end up with after that recombination was complete is a sister chromatid that now has genes A, B, G, and H, right? And another non-sister chromatid over here that would have genes E, F, C, and D. And so what we've seen here is what's referred to as a translocation of genetic information. So genes that were supposed to be present on this chromosome have now been moved or translocated to another chromosome in the genome. And so this also represents a deletion, because this sister chromatid is now missing genes C and D. And again, remember, where are these sister chromatids, or sorry, where are these individual DNA molecules going to end up? They're going to end up in different gametes. So whichever gamete inherits this particular chromatid is going to be missing genes C and D on this bit of DNA. Now they probably are going to be inheriting this other chromatid over here that has those genes, but it's still going to be a set of C and D down. So when there's an error in recombination in which non-homologous chromosomes are paired up and they recombine or exchange genetic information, that's going to result in a translocation and deletion of genetic information. And again, the consequences of that mistake are going to depend very much on what those genes actually encode. What are the proteins that are produced as a consequence of those genes being expressed, and what are the consequences of not having that protein or having too much of that protein in the cell? All right, so how does this play out in terms of evolution? Well, we can see a really nice example of how this sort of gene duplication event can play out in an evolutionary sense, because it turns out that once you start having these duplication events, you can start to see the evolution of what are called gene families. So families of genes that have related functions that are all descendant from one original ancestral gene. And the way to think about this is like so. Let's say that you have that chromosome and it's got genes A, B, C, and D on it. And let's say that gene B is an essential gene, right? You absolutely have to have the function of whatever that gene encodes. And if you make mutations that change the function of that gene, then that's a lethal mutation and the organism cannot survive that. But now let's see what happens after that gene duplication event, where now you have 2 copies of that gene, right? As long as one copy stays with its normal sequence and continues to encode the normal gene product, that organism is going to survive. And that second copy that's the result of the gene duplication event is now free to undergo mutation, which happens all the time, and to possibly acquire new functions, right? So gene duplications result in extra copies of genes, and so even for essential genes, we can start to see evolution of proteins with different functions. And if those proteins become different enough, we can actually see increasing complexity in terms of physiology. If we see isolation of populations with those different genetic sequences now, over time that can actually lead to speciation, where organisms become different enough from one another, and populations of organisms become different enough from one another that they actually become separate species. All right, so how is this really nicely illustrated by the globin gene family? Well, if we go back in time about, 800 million years ago or so, there would be the common ancestor of the globin genes. And the globin genes, as we've talked about before, encode the protein subunits of hemoglobin, which is that protein complex that carries oxygen around in our blood. So if we go back 800 million years ago, what we would see is one ancestral globin gene in the common ancestor of all vertebrates, and actually all vertebrates and invertebrates. If we look at the globin genes that are present in the human genome today, what we would see is that there are 2 separate clusters of globin genes. What's referred to as the alpha globin gene cluster, and that's on chromosome 16 in the genome, and the beta globin gene cluster, which is on chromosome 11. And if we looked more closely, we're just going to look at the beta globin gene cluster just for the sake of simplicity. What we would see is that there are 1, 2, 3, 4, 5 globin genes in this beta globin gene cluster that are functional, that actually encode functional proteins, and then one what's referred to as a pseudogene that does not encode a functional protein because it has undergone mutation that has destroyed the coded protein's function. So when this gene gets transcribed, it doesn't encode a functional protein anymore, right? But all of these other copies do. Well, how did we end up with so many copies of the globin genes? What's hypothesized to have happened, and what the molecular evidence suggests, is that at some point there was a duplication event, just like we just talked about. It could have occurred during recombination error in meiosis, and so we ended up with 2 copies of that ancestral globin gene. Over time, those 2 copies diverged from one another, and so one copy perhaps started to accumulate mutations, changes to its sequence that caused it to have slightly different protein sequence and therefore slightly different structure associated with it. And so that would have been the ancestral gene to the alpha cluster and the beta cluster. And then a translocation event could have separated those 2 genes and put them on different chromosomes, right? And then further gene duplication events occurred over those hundreds of millions of years of evolution, leading to these clusters of genes that have highly related sequences but may differ very slightly in their functions. And so what do I mean by differing slightly in their functions? They're all globin genes. They all encode globin proteins that are part of the hemoglobin protein complex. But it turns out that these 2 gamma globin genes that are associated with the beta globin gene cluster are fetal versions of hemoglobin that are only expressed during fetal development. And they actually have a slightly higher affinity for oxygen than the other adult hemoglobin versions that are encoded by the other highly similar gene sequences. And because of that slightly higher affinity for oxygen, the fetus actually has an advantage in survival in being able to acquire oxygen and uptake oxygen and hold on to oxygen even when oxygen is at slightly lower partial pressures, which is the situation that we see in the human placenta. So where the fetus is acquiring its oxygen from, there's actually a mixture of fresh oxygenated blood and deoxygenated blood, and so the concentration of oxygen is a little bit lower in that tissue. So the fetus is actually requiring a hemoglobin protein that has a higher affinity for oxygen so it can bind oxygen at those lower concentrations. So this gave fetuses a very definite advantage, and so that's how we could see a selective pressure to maintain those slightly different globin genes that encode globin proteins with slightly higher affinity. So this is a really nice example of this evolution of a gene family, in this case the alpha globin genes and the beta globin genes, all descended from a common ancestor over hundreds of millions of years of evolution, acquiring slightly different functions that are associated with selective advantages. So gene duplications aren't always a bad thing. They can be associated with speciation and with the evolution of genes with new functions, and therefore increasing complexity of the organism that we're talking about. Now, up until about 15 years ago, it was thought that most of the human genome, if we looked at different humans in the population, was actually pretty similar, and that maybe what we would see is, maybe 0.1 to 0.4% of the genome varying, and the way that they would vary is in terms of what are called single nucleotide polymorphisms. That is, individual nucleotide base pairs that varied in their sequence because of random mutations that are introduced all of the time. Right? And so before the advent of high throughput, more modern genome sequencing technologies that became available over the last 10 to 15 years, that was pretty much what was thought to be happening with the human genome. If you compared one human's genome to another, you'd see single nucleotide changes in sequence between those two genomes. But now that we can very easily sequence thousands and thousands of humans, what we see is that there is a considerably greater level of variation than we ever thought possible. And more specifically, the type of variation that we see is in gene copy number. So not just those slight changes to DNA sequences as a result of mutation, but also duplications and deletions that have led to significant variation in how many copies of particular genes a given individual human might have. And if you look across the genome as a whole, this is a diagram that's showing all of the autosomes and sex chromosomes in one complete set of genetic information, while showing slightly more than one set because it's got both of the sex chromosomes. What these lines are indicating is positions within the human genome where this kind of gene copy number variation has been demonstrated to exist, right? And so what level of variation are we talking about? Well, it turns out that if you compare the DNA sequence of two individual humans, that also is at about 0.4% of the genome, right? Where I might have at a particular position in my genome three copies of a particular gene, and you might have at that same gene locus only one copy of that gene, right? Now, as you can imagine, depending on the gene, that's going to lead to differences in physiological function, differences in metabolism, and potentially differences in disease risk. And there are a number of human diseases that have been linked to this sort of copy number variation. It's been particularly well demonstrated in human cognitive disorders and psychiatric disorders, things like schizophrenia and autism, which seem to have a common underlying basis in developmental processes that go on in the central nervous system. So 0.4% might not sound like a whole lot of DNA, but remember that the human genome is 3 times 10 to the 9th base pairs long. So 0.4% of 3 billion base pairs of DNA is a significant amount of variation. Okay, so errors in recombination can lead to both duplications of gene sequences, which can be associated with disease risk. It can also be associated with evolution of new functions of genes and genes families. It can also result in translocations. What I want to talk about next are the other type of errors that we see happening in meiosis, where there's a failure at the spindle checkpoint that regulates the progression from metaphase to anaphase. We already talked about that in the context of mitosis. The very same checkpoint operates in meiosis. It's slightly different obviously, but same idea, making sure that all of the chromosomes are correctly lined up, are correctly connected to the spindle apparatus before allowing the cell to proceed from metaphase to anaphase. So if there's a failure of that spindle checkpoint and it doesn't work correctly, and it allows chromosomes to prematurely be segregated before they're correctly hooked up to the kinetochore microtubules, then what we see is non-disjunction, a failure of correct segregation. And so in the context of meiosis, this could happen in meiosis 1, where we know that normally homologs are being separated, or it could happen in meiosis 2, where we know that normally sister chromatids are being separated. So let's take a look at what would happen in either of those scenarios. So here on the left, we have an example of a meiosis where the non-disjunction event occurs in meiosis 1. So here we see anaphase, where this tetrad, right, this set of duplicated homologs, is being correctly segregated, one homolog being sent to this pole of the cell, the other homolog being sent to that pole of the cell, but this tetrad is not being segregated correctly. There's a non-disjunction event, a failure to disjoin, where this duplicated homolog failed to go in this direction, and both end up going in this direction. And so what we get at the end of that first meiotic division is a cell that has one too many of this set of duplicated homologs, right? So it inherited too much DNA here. And this cell, which didn't inherit any copies of that duplicated homolog set, so it's missing genetic information. If these two cells then undergo that second round of meiotic division, where sister chromatids are now separated from one another, then what we end up with is half of those products of this meiosis that are n+1, meaning they have one too many chromosomes of whatever that chromosome type was, and half of the gametes produced will be n-1, where these are gametes that are missing that entire set of genetic information. Now, what's going to be the consequence of that? Well, it depends on what that chromosome is as to whether that's going to be tolerated or not. Most would not be tolerated. So if there's a failure of segregation in meiosis 1, all of the gametes are going to be abnormal. Half will have one too many chromosomes, and half will have one too few. What if the failure to segregate occurs in meiosis 2, however? What we would see is meiosis 1 proceeding normally, duplicated homologs being separated as they should, being sent to opposite poles of the cell, right? And then let's say that this cell undergoes a normal meiosis 2. So nothing untoward happens in meiosis 2 in this cell, and what we get are two gametes that are entirely normal and haploid. They have one of each type of chromosome that they're supposed to. But let's say that this cell has a failure to segregate one of its chromosome sets correctly. And so these two sister chromatids, instead of being separated in meiosis 2 as they should be, end up both going to the same pole. And again, this would only happen if there was a failure of the spindle checkpoint. Then what we would end up is two abnormal gametes, one with too many chromosomes, one with too few. So one n+1, one n-1. So in this scenario, if the failure to disjoin is in meiosis 2, then half of the gametes are abnormal, one n+1, one n-1, and half of the gametes would turn out to be normal. Now, when you have a non-disjunction like this, a failure to disjoin, and you end up with too many or too few chromosomes, that's a defect in chromosome number. And so that leads to something called aneuploidy. So remember that ploidy refers to chromosome number. U means true. So aneuploidy means not true chromosome number, right? So whatever the normal chromosome count is supposed to be in these gametes, it's off by one or more. What would be the consequences to a human being that had this sort of non-disjunction event happen in the gamete from which it was derived? Well, we can see that most autosomal aneuploidies are actually lethal and not survivable. It turns out that if you look at human pregnancy and you look at the rate at which a given human pregnancy is going to survive even past the first trimester, what you find is that about 20% of pregnancies spontaneously abort in the first trimester, and a huge percentage of those actually fail in the first 2 weeks before the woman even knows that she is pregnant. So there's a huge failure rate associated with the beginning or onset of pregnancy, and almost all of those, 90% of those spontaneous abortions, are associated with fetal chromosomal abnormalities. So there's some sort of aneuploidy or other chromosomal abnormality that prevents that fetus from making it further, or sorry, prevents that embryo from making it further into fetal development. So most autosomal aneuploidies are going to be lethal, right? They're not going to be survivable. You're not even going to make it to a live birth, much less make it into adulthood. There are 3 that are actually seen a bit more often and do make it to live birth. So these are trisomies. So this is referring to 3 sets of a particular chromosome. So 3 chromosomes instead of 2, right? And those are chromosomes 21, 18, and 13. And the incidence of these trisomies for chromosome 21, about 1 in 800 overall, about 1 in 5,000 for chromosome 18, and about 1 in 10,000 for chromosome 13. So here on the left is the normal human karyotype that we looked at before, where we see the paired duplicated homologs for all of our sets of autosomes 1 through 22, and then 1 set of sex chromosomes X and Y. And what you see here on the right is a karyotype that's depicting a trisomy 21, where this individual actually has inherited 3 sets of chromosome 21 particularly. And this is associated with a not infrequent human genetic disorder that is Down syndrome, right? Which we're all familiar with. So Down syndrome is associated with a cognitive disability. It's also associated with various defects in development of other organ systems, including the cardiovascular system. Kids born with Down syndrome very often have holes in their heart that have to be repaired, or other heart defects that have to be repaired after they're born. It's also associated with defects in the digestive tract. So they have a problem with getting adequate nutrition and have to account for those difficulties in obtaining nutrition in various ways. If you look at the incidence at which Down syndrome occurs as a function of maternal age, you actually see something very, very interesting. So here on the y-axis is incidence of trisomy 21 per 1,000 live births. So this is babies born. And down here on the x-axis is maternal age. So going from onset of puberty about age 15 on up to age 45. So what you see is that at about age 25, up to about age 25, incidence of Down syndrome in these moms is pretty low, about 1 in 500-ish. But if you look at the incidence of Down syndrome as mom's age goes up, then by the time you get to age 45, what you see is that the incidence of Down syndrome has gone up hugely and is about 1 in 25. Now, to understand why that happens, you have to understand a little bit more about how the meiotic divisions that produce the maternal gametes happens in humans, right? Because it's a little bit weird. What happens is that the entire set of gametes, oocytes, is actually produced during fetal development of that human. All right. So for every human female, her entire set of eggs was actually established when she was still a fetus in her mother's womb. All right. So all of the primordial germ cells, which generate those oocytes in the female fetus, initiate meiosis before birth. All right. So that meiosis stalls at prophase 1. So in every one of those primordial germ cells that have initiated meiosis, they're stalled at prophase 1 with the duplicated homologs all paired up, right, in synapsis, held together, and then that meiotic division just pauses. It stalls and doesn't go any further, right? So then this fetus continues development. It's, that baby is born, it goes through childhood, it goes through, early adolescence, and then beginning with puberty, at every menstrual cycle, there's an ovulation event. And at each ovulation, the oocyte that is being ovulated picks up and initiates meiosis from where it stalled back here, during fetal development. And so here, this meiotic division is actually picking up, and we see meiosis 1 completing. Right? So here's ovulation. Here's that oocyte being released from the ovary. Here's the ovary into the fallopian tube. And as it travels down that fallopian tube, if this is ovulation that's going to result in a fertilization by a spermatozoa, only then will that oocyte actually complete the second meiotic division and resulting in a fully mature oocyte that then is going to fuse with that sperm. All right, so it's a really complicated meiotic division that human females go through. So a human female that's 25 years old and ovulating an oocyte that is then going to be fertilized by a sperm, that oocyte actually began meiosis 25 years previously when she was still a fetus in her mother's womb. So now imagine you're a human female that is 45 years old and you're ovulating, and that's going to result in a fertilization, right? That oocyte initiated meiosis 45 years ago. So now what we're asking is for this very complicated molecular process that was initiated in the fetus during fetal development and has all of these proteins that are, connecting these tetrads up and the kinetochores and all that good stuff. We're asking that whole molecular complex to remain intact, and then at ovulation to pick up and, begin where it left off and, reinitiate meiotic division and go from there. And so, as you can imagine, proteins degrade over time, and this molecular complex could fall apart in any number of ways. And so when we think about this increased incidence of trisomy 21 as a function of maternal age, that's because there's a huge increased incidence of non-disjunctions or failures to disjoin as a function of maternal age in human females. Now, the molecular details of what exactly goes wrong and what happens to this protein complex that's holding these together and why don't they segregate appropriately the way that they're supposed to, that is not entirely understood. We don't know the details of why that happens. We just know that it does, right? So you might ask yourself, well, does the same thing happen in human males? Is paternal age actually associated with any sort of increase in genetic defects and genetic disorders in the offspring? And the answer is maybe, but not in the same way, all right? So to understand that, we then have to look at how a spermatogenesis occurs in human males, because it's different. So whereas human females get their primordial germ cells and their whole collection of oocytes established before birth, so they have this entire pool of about 400,000 immature oocytes in the ovary when that baby is born, and that's all they're ever going to get, ever. And then those oocytes, reinitiate meiosis at ovulation and then only complete it at fertilization. That's the situation in females. In males, it's very different. So males start with a primordial germ cell and expand that out to a population of stem cells that exist in the testis, right? And then at puberty, those stem cells undergo characteristic sets of division to renew and expand the population of stem cells. And as each of those stem cells undergoes a differentiation event, leading to mature spermatozoa, each of those cell divisions is associated with a DNA replication event. And every time DNA gets replicated, there are mutations that get introduced into the genome. So when we're thinking about this stem cell population that maintains this set of cells that are constantly capable of dividing and producing new spermatozoa, millions and millions of them every day, that those cells actually replicate and divide about 23 times a year to renew this population of stem cells. And then as those stem cells undergo divisions to produce the mature spermatozoa, that's associated with another 4 replication events, right? And so over time, as each spermatozoa is produced, that could be the result of hundreds of previous cell divisions that produce that mature sperm. And at each of those cell divisions, each of those rounds of DNA replication is going to introduce new mutations in the form of single nucleotide base pair changes in that genome that is the genetic payload being carried by that sperm. So if we compare the level of mutation that's present in a sperm to a level of mutation that's present in an oocyte, in sperm we see many, many, many more mutations that are apparent, right? Because once these oocytes are established in female, they don't divide again. So throughout the adult female's life, those are not undergoing additional rounds of division. Only in the male do we see that happening. And so the sorts of genetic disorders that you see being linked to a higher prevalence in older fathers are the result of those kinds of single nucleotide base pair changes. And so there are disease syndromes like achondroplasia and Apert syndrome that have very well established associations with increasing paternal age, where you can see that as dad gets older, those disorders become much more likely to occur in those, and that's a result of mutations in specific genes that are particularly sensitive to mutation effects. And there are other disorders that are associated with increasing paternal age as well. So autism has been linked to increasing paternal age. There's some suggestion that schizophrenia is also impacted by paternal age as well. Okay, but aside from trisomy 21, which is relatively common, other autosomes and extra copies of those autosomes are not well tolerated as aneuploidies. And, you know, the explanation as to why, we have to think about how many genes are on the autosomes and, you know, what types of genes are on the autosomes. So this chromosome number 1, for example, is humongous and contains 4,500 individual genes. If you inherit an extra copy of chromosome 1, you are dead very early in embryonic development. That's just too many genes in extra copy to be tolerated during embryonic development, where there are going to be lots and lots of genes on that chromosome that are going to affect development in an adverse way, right? But you can also have other chromosomes that are smaller, don't have as many genes, but may have very, very important genes that cannot be tolerated in too many copies or too few copies. So in other words, with gene dosages that are altered from the normal gene dose that would occur having 2 copies of that gene in the genome. Well, what about the other chromosomes aside from the autosomes, the sex chromosomes? It turns out that aneuploidies of the sex chromosomes are much more common and are better tolerated in terms of physiological function and development. All right, so if we look at the karyotypes that are associated with sex chromosome abnormalities, we can see that a couple of the most common are where we have 1 extra copy or 1 too few of the X or the Y. And so this first one that's listed here is something called Turner syndrome, where the individual only has 45 chromosomes, so it has 1 X, but it's missing that extra chromosome, either an X or a Y, and so that's often referred to as being XO. And the incidence of that is about 1 in 2,000 female births. And so if we look at the types of abnormalities that are associated with that, there aren't a whole lot. They don't have really cognitive disability. They don't have a whole lot of behavioral disorders associated with that. They do have a slight defect in gonadal function, so reproduction is impacted in these individuals, and they have some other kidney and cardiovascular defects that are associated with that, but not too severe, and they definitely survive past birth and into adulthood just fine. XXY, this is a syndrome that's referred to as Klinefelter syndrome, and that occurs in about 1 in every 576 males. In terms of defects, a little bit of cognitive disability, a little bit of behavioral changes that are associated with that, taller than average, again, hypogonadism, so not exactly normal fertility, and then various other disorders in other organ systems that are associated with that. And then there are other either trisomies where we've got extra copies of the Y or the X occurring in various frequencies, and then additional aneuploidies where we can have even tetraploidies or pentaploidies, where you've got 4 copies of a sex chromosome, 5 copies of sex chromosome, of unknown incidence, certainly quite rare. We're into the tens of thousands to hundreds of thousands of male or female births here. Right, so why are these aneuploidies better tolerated? Well, 2 things. Number 1 is that it turns out that in order to compensate for the fact that females have 2 X chromosomes and males only have 1 X chromosome, what typically happens is that chromosomes in excess of 1 X chromosome get inactivated during development. So random inactivation of all but 1 X chromosome happens typically in human females. So if you have extra copies of X's, then those extra copies just get inactivated, essentially. It doesn't work perfectly, and so that's why we see some disorders associated with having those extra X's. But certainly the cells in that female fetus try to shut down those extra X's. As to why extra copies of the Y can be tolerated, Well, it turns out that there aren't any essential genes on the Y, and that makes sense if you think about it, because half the human population doesn't have a Y chromosome, so there can't really be any physiologically essential genes on the Y. It has one very, very, very important gene, and that's the gene that confers maleness, SRY, that we'll talk about more in the genetics discussions. But in terms of essential physiology, there really aren't any. There are only about 200 genes on the Y chromosome, and only about 70 of those actually encode proteins. So very, very, very few genes, none of them with really essential physiological functions. So if you have extra copies of them, not really a big deal. So trisomies that are associated with extra copies of the Y are also very well tolerated, right? But those are sort of the exceptions to the rule. Most chromosomal abnormalities are going to be involving the autosomes, just because there are 22 of those and only one set of the sex chromosomes, and most of those are lethal and not tolerated well at all.